Go. It says record it now. And yeah, so if you want to remain anonymous, then go ahead and just turn your video off. And our YouTube channel is called NSOF Wash DC, and you can watch all of our previous meetings during the pandemic. We had them all on Zoom, and we recorded them, so it's great. So it's really nice. It's great to go back to them. Hey, so and we can put the link on the chat there. The next order of business is reading and approval of the minutes. And I did see Mr. Secretary Gary Hevel. Will you please read the minutes from the last regular meeting?
The 1230th regular meeting of the Entomological Society of Washington convened at 7 p.m. on the 6th of October in room WG33 of the National Museum of Natural History, with Arist Lines presiding. There were 50 members and guests in attendance, the majority joining the meeting virtually by Zoom. President Chamorro noted that this is the first time during the pandemic that such a joint meeting has occurred. The minutes of the May meeting and the annual banquet were read by Recording Secretary Gary Hevel and approved. President Chamorro announced that Alyssa Seaman has accepted the position of curator for the society, replacing Nick Silverson, who has returned to academic studies. President-elect Matt Buffington announced that he has found members for the auditing and nomination committees, and that candidates for officers will be presented during the November meeting. Membership and Communications Secretary Elizabeth Young was not able to attend the meeting, but left the following names to read as new members: Michael Thoreau, Sonia L. Petroza, Patrice Mellon, and Michael Pesdapula. President Chamorro announced that the Society is redoing our website with use of a program called Squarespace. The new system provides many advantages for storage, adding new data, customizing merchandise, and keeping track of memberships. A sample page was displayed on screen that showed how members can develop their own information on their entomological interests. Members will gain access through Bio1. During the past year, Society officers have been developing a special issue saluting Ray Gagne. The issue shown on screen will soon be available and contains articles from friends and associates that honor Ray. Program Chair Alan Norbaum showed specimens of tephritid flies that he collected in the Great Smoky Mountains recently. Ruthie Pearson from Washington State discussed her purchase of a nest of the northern giant hornet, adding that she had arranged for 3D printed plastic replicas of the nest, which allowed children to feel and observe the object. Alma Solis passed around a specimen of a pyralid moth that was recently found in Detroit and had not been seen since the description of a single specimen in 1912. The original specimen was from the Philippines, and the new find in the United States attracted strong interest from the news media, including CNN and the New York Times. Matt Bertoni was the speaker of the evening and was introduced by program chair Alan Norbaum. The title of the presentation was Adventures with Arthropods, Extension Diagnostics of a University Entomologist. In his position, Matt meets with a variety of people through phone calls and visits from North Carolina citizens commonly identifying insects. In 2021, he had 3,000 such requests. He has 8,000 followers currently on Twitter. Brown recluse spiders have been a hot topic recently, and they are rare in the state. Hammerhead and jumping worms have also been of consumer interest and have been in North Carolina for decades. Matt is also an insect collector and has a collection of over 1,700 species. His interest and expertise in photography have led to working with areophyid mites, and he has found 4 new species in this group. Matt enjoys this position and points out that North Carolina is a great place for study. The state has a varied topography with sandy shores on the east, a mountainous interior, and areas in the west to allow production of grain. Throughout his presentation, Matt showed examples of his amazing microphotography successes. The meeting is adjourned at 8:30 p.m.
Very much so, Mr. Secretary. I'd ask for a motion to approve the minutes as presented. One second. All those in favor?
Aye.
Opposed? Okay, motion passes. And we love your little doggy, by the way.
You always go there.
That was great. Okay. So now we move on to reports of officers and committees, and I'd like to ask Matt Buffington, President-elect, to present what was promised.
Should I do it in front of Alan's computer?
No, do it
Oh,
here, because
okay.
he's
Oh,
muted.
this one,
So,
okay.
let's see. You're right.
Okay, cool. Hi, everyone. So I'm president-elect of the Entomological Washington, and part of my job is to have the slate prepared for the next group of officers to be voted in. My committee successfully executed my request, and everyone has agreed to stay in their positions. The one position that is going to change is that of president-elect. As I become president, we'll get a president-elect, and that will be Dr. Dawn Weber from Beltsville Area Research Center, USDA, who also co-wrote the most recent massive tome on Charles Valentine Riley. And since C.B. Riley co-founded, organized the society, it is fitting that Dawn Weber be, I think, president-elect. I think it'd be really great. I think, is that the other?
She just has the formality.
But Havel already announced her.
Yeah,
In the
for the
minutes.
election, so it will be voted.
Right, okay. So, sorry, Alyssa will be voted in as well. She'll be. Yeah, I guess you didn't really agree to stay in position because it's a new position. My apologies.
Yes, the email.
That was like,
Do I have to announce the whole slate? I think so.
yeah, go ahead. I guess.
Is it written down?
Yeah.
Oh, it's right here.
Yeah. so just
Okay.
change.
Okay. So the program chair will remain Alan Norbaum. Thank you, Alan. So Alyssa is now curator. Okay, thank you, Alyssa. Curator. Recording secretary will remain Gary Hubble. Membership and communications secretary will remain Elizabeth Young. Treasurer will remain Abby Kula. And editor of our prestigious journal will remain Mark Metz. Did I miss anyone?
I think technically these people are being nominated to
Right,
serve again next year.
okay.
And then December, there's a vote.
I think I get this right.
That's the way it works.
So, and
Well, I'd
then
like
there'll be a
to
vote.
formally nominate. the
Nobody
people I
votes for
just
me.
read.
Nobody votes for me, then I guess I won't be program chair. We only have
All
one
of
person
them.
for every position.
Okay,
It's kind of a
okay.
shoot.
Okay. Well, I move that those be the nominated slate and we will vote on them next meeting. Secret ballot, right? Just want to make sure.
I'll say.
Okay, that's a second. Very. Okay.
Do I get to do the gavel? No, that's your thing.
Thank you.
Gotta wait one more month.
All right, one more month. Do it.
Okay, thank you very much, Matthew Buffington, for that. And now we move on to Media and Communications Secretary, Elizabeth Young. So if you want to come over here. And it's also an opportunity for introduction to new members and visitors as well. So, okay.
Good evening, everyone. We have one new member to announce for November. Their name is Hazel Hawk. And we have 4 official members who were announced in October. Michael Farrell, Sonia L. Pedroso, Beatriz Rodriguez Velez, and Makoto Takuda. And if there are any guests today, either over Zoom or in person, please, we welcome you to introduce yourselves, say hi. Love to hear from you.
Yeah. Okay, we have.
Hi.
Hi, I am Giovanna Caso. I am a Mexican scientist, and I am a new fellow here, working with Sean Brady and Robert Kula with Baconics. So yeah, I am still like in the first step for developing my project, and yeah. Hello to everyone.
Hi,
Welcome.
welcome.
Thanks for coming. Thanks.
Can everybody hear in the Zoom call?
Yes, anyone on the Zoom call, any guests, visitors, feel free to unmute yourselves and introduce yourselves now. Click.
Oh, we have another visitor here.
Oh, yes. Hi, hello, yeah.
Hello, my name is Andrew Jansen. A new hire at SEL for the electronic confocal microscopy unit. And yeah, I guess this will be my first time here. Hopefully first of many. Thank you.
Welcome, thank you for coming. Okay. Great. Okay. Thank you so much for coming, everyone.
Thank you, Elizabeth.
Thank you.
Hey. So now we move on to unfinished business, and my only thing was to say that the website is still ongoing, but you should be receiving some notifications in the coming weeks as we're moving closer to it. First, we're going to try with the executive committee to get it running and get all the glitches figured out before it goes public. So be on the lookout for that, and we're timing it exactly so that you can renew your membership with the new website. So. Okay, any new business? No new business. And now we move on to presentation of notes and exhibition of specimens. So we have a few books that I know Richard Green brought with him. Yeah, and Matt Buffington as well. So if you wanted to come over here so people can see you if you're not too shy, you can move over here, and then this actually captures.
Is this gonna take a while?
No. I'm gonna read through.
Make sure recording secretary Hubble records everything you read.
No, I'm not gonna mention. I should have brought these out. This is a sampling of things that have been acquired directly through library acquisitions and items that I've donated and other people have donated to the entomology library. They're old and new, but none are currently within the library. So there's several books on archaeoentomology, if that's actually a word, and one on paleontology, the insect evolution.
Around.
So archaeology and entomology is on the Eastern Mediterranean, which is sort of interesting. And then there are 2 books from British Archaeology Society on insects and found in various relics. So nice massive tome and by Richard Wilkinson, who works for the Walter Reed unit. So this is basically a collection of mugshots of mosquitoes. So 2 volumes are very, very colorful and insects of Japan, which is actually this is all weevils.
All right.
We'll take that one.
And probably the most charismatic insect in the arachnid world, ticks.
Oh.
Europe and in Africa. Actually, they're only black and white mugshots in here, but basically it's a catalog, a descriptive catalog. And Scott Miller donated a book on, seems timely, on vampire moths. So this was published in, I believe, Thailand. It was a pain for him to get. Yeah, Thailand. And there are some other items here which people can take a look at. I don't want to belabor.
Okay. Very nice.
There's a new journal from France on Lepidoptera. An old publication from the 1920s, the Entomological Fauna of Indochina, which was published between 21 and 25. Some of these are online, some of them aren't. Most of it's coleoptera. We got this one.
And then you have a giant ornithoptera book and.
Yeah, this is on bird-wing butterflies, mostly of Java, Burnet moths, natural history, with really fantastic photos of adults, and caterpillars, and the eggs and the vegetation that they're found on. 2 volumes set. So, if anyone's interested, I'll wheel them over by the groaning board here.
Well, we had a backlog, so it makes sense. Yeah, thank you very much for that. And Matt Buffington has a couple of books he wants to share as well.
So Loris and I were in Stockholm over the summer, and friends of ours gave us some fantastic Swedish entomology books. One on scarabs. Oh no, dung beetles of Sweden. Yeah, fascinating. My buddy Matthias is one of the co-authors on this. Each species, and they have a plan in Sweden that every single species of insect will have a common name. Every single one. So all the common names for the dung beetles, if you're interested in what the Swedish common name for a dung beetle is, it's in here.
Great. Thank you.
The other one, we got a colleague of ours to these little kind of guidebooks. This one's on Hoppspindlar, which are the jumping spiders, which is such an awesome name. Yeah, makes me think of going to IKEA and buying furniture. Then grasshoppers, moths, odonates, butterflies, and mosquitoes. So if you want to just take a look at these kind of nice little novelties, are so well done. I mean, I think the Swedes know a thing or two about classification. So
thank you very much. And then I think we might have some announcements as well from our Zoom attendees. So yeah, someone be giving an ECA announcement or ECN announcement. So Jill, if you wanted to go ahead and do that. You can go ahead.
Hi. I'm sorry, I can't do it. I was asking if somebody else was going to do it.
Well, the ECN meeting and the ESA meeting are coming up. And that, the Entomological Network meeting and the Entomological Society of America. So be on the lookout for that, for those meetings. They're very relevant for our, I don't have the dates off the top of my head. Okay, so now we move on to our presentation. Thank you very much for your patience, Jeff Skevington. And I now cede the floor to Alan Norbaum, who will be presenting our featured speaker. So why don't you come over here, Alan? Because if you unmute yourself. Oh, really? Okay, yeah, bring yours.
Hi, everybody. Before I forget, just tell Gary, we had 37 participants on Zoom. We are about 15 or so here. Elizabeth's going to pass the sheet. And our December speaker is tentatively Dave Adamski. And so now I have the pleasure to introduce our speaker tonight, who is Jeff Skevington. Jeff has been a research scientist at Agriculture and Agri-Food Canada for 20 years. He is also an adjunct professor at Carleton University. His research focuses on the taxonomy, phylogenetics of flies, particularly flower flies and big-headed flies. He's published over 100 research papers and books on these insects. Jeff is a lifelong naturalist with an interest in all aspects of natural history and has a soft spot for birds as well as insects. So no further ado, we need to share the screen.
Yeah, so let's do. Let's go ahead and share your screen, Jeff, and then we'll mute ourselves, okay?
Okay, all set. Thank you very much, Alan. Is this coming through loud and clear for everybody? Good, seeing lots of nods. All right, so I'm Jeff. I'm from the Canadian National Collection of Insects in Ottawa, and I was considering doing more of a research talk, but this is like our meeting tonight. It's a hybrid, so I'm going to give you some general natural history on Syrphids and then tell you a little bit about some different research projects, just glazing over and touching on a few different things. So hence the title, Syrphids and Other Stuff. So let's see, there we go. Most of you, I'm sure, know how to recognize Syrphids more so than many of my audiences do. Although you may know to look for the spurious vein based on key features, they're really, really easy to recognize based on features of the face and the head. If you're not sure if it's a wasp or a bee or some other fly, you can usually tell it's a Syrphid just by looking at the head. You know, there are exceptions. The middle one at the top, there's Toxomers, and it's got long antennae and sort of breaks the rule. As you know, doesn't look like a lot of Syrphids because of those long antennae. It looks more like a strat. But anyways, they're generally pretty straightforward to identify, and many mimics in the group. Mimicry is one of the things that really people think of when they think of Syrphids. In fact, as I've been talking about Syrphids for the last few years to general public, it's amazing that most of them actually think that they're bees and wasps. They just they're so similar. The mimicry is so good. So I've just thrown a few things on here. On the top left is yellow jacket, and below it is Chrysotoxum, one of the Syrphids that mimics them, and below that's a species of Spilomyia longicornis, that is an excellent mimic of yellow jackets. On the right is bald-faced hornet and its Syrphid mimic, which is fantastic. Spilomyia fusca is below that. So a group that's full of mimics. And in fact, some of the things that you wouldn't think are mimics, you look and they're little black flies, and then you watch them fly and you realize they look just like a lot of the little black bees. So mimicry is almost throughout the entire group. So it's so profound that in fact, most people seem not to know what Syrphids are, whether it be the media or even editors putting books together. And so it's always fun, and we always collect these. You can see that these are all Syrphids, of course, right? They're not bees. Wouldn't you love to write a book on bees of the world and then have somebody use a stock photo of a musset and put it on the cover? Anyways, so that, you know, it's not just Syrphids that are mistaken. There are a few other things thrown in now and then, but Syrphids seem to be commonly mistaken, especially in the common media. Whenever there's a call for a bee photograph, it seems inevitable that they use a Syrphid. So it's funny. So let's talk about mimicry first. It's amazing how many Syrphids are what we call perfect mimics, that they're really, really difficult to tell apart from their models. So here's a bunch of perfect mimics shown here. Cryorhina on the top left, which is a bumblebee mimic. In the middle is Arist tenax, common dronefly, which is a honeybee mimic, et cetera, et cetera. So these are perfect mimics. But we also have these things that are called imperfect mimics. They converge on a generalized model, but not anything specific. And that's what I would say are the hard to identify syrphids that all sort of are yellow and black and often require a little bit more scrutiny to identify. It's those perfect mimics are usually pretty dead easy to identify. They're the birds of the fly world for sure. I thought I'd throw in this cool example I found online with a wasp on the left and 2 syrphids and a soldier fly, all mimicking the same wasp gestalt. So there's been a lot of research on syrphids from a mimicry angle because of this, and that's been a lot of fun. I've worked with various people on mimicry as a result. It's broadened and enlightened my research. So normally I'd do this in person and I'd get you to all shout out what you think. I suppose you could use the chat, but I don't even see where the darn chat is here. So anyways, you can think to yourself, how many syrphids are in this photograph? How many syrphids are there? And how many are bees and wasps? I'll give you a minute to count. So this was a picture that one of the students I worked with that was studying mimicry used when she was doing surveys of people to try to get their ideas of what was a bee, what was a wasp, and what might be a mimic. And almost nobody gets it. There are 7. So top row, the 2 right insects are syrphids, even that right-hand one. And that's really cool. You know, anyone who works on flies knows that fly antennae are really follow a very, very tight genetic pattern. And so not many syrphids have long antennae, which most wasps and bees have. So this one, it's actually an outgrowth of the face that makes it look like long antennae, and the little antennae are perched on the end of that one in the top right. It's one of the Syriana. So 2 in the top row. The next row, it's 3rd from the left and the right-hand one. So the right-hand 2 again are syrphids. Bottom row, we've got syrphid, and honeybee. In the bottom right is honeybee. So 7. Okay. So in terms of diversity, this is described diversity, so it's not real, right? I mean, but this is what we know in terms of described species. So flower flies are right up there in terms of fly diversity. With over 6,000 species, they're one of the most diverse living groups of flies. There are really underrepresented groups like gall midges, right, which maybe have 100,000, maybe even 1 million species, who knows, right? But or tachinids, for example, which bristle flies they're called here. I give this talk or parts of it to audiences who like common names often. So bristle flies are Tachinidae, with nearly 10,000 described species and undoubtedly 100,000 really. But flower flies are pretty diverse. We think that there are certainly over 10,000 species in the world. So still lots of work to do to describe them all. In Canada, there are 537 species. In the US and Canada combined, 860. And of course, we're always tweaking that as we find new things. And amateur naturalists are finding a lot of new records now. This is something that's fairly novel. You know, 10 years ago, it was just research scientists working on groups like this. But now iNaturalist has opened the door, and tons of new finds coming in from people using iNaturalist in particular. So pretty exciting for any of us working on groups that are kind of like the birds of the insect world, if you wish. They're more readily accessible by the populace overall. So I wanted to give you a quick snapshot of some of the different natural groups or groups of syrphids. They're not all natural. So this is Surfinae. These are mostly imperfect mimics, yellow blacks like this. I'll talk about the natural history of these groups a little bit later, but that gives you a bit of an idea of the gestalt. Another big group of syrphids are the Aristallinae, which is not a natural group, but we're about to fix that in an upcoming paper. And then the Microdons, or ant flies, are another natural group of syrphids that are not flower visitors like all the others. And I'll talk a little bit about their natural history in a minute. And the last group of those black ones, Pipizinae, which are root aphid predators. And we'll talk about them briefly as well. So a little bit more about natural history of these groups in general then. So when I started working on syrphids 20 years ago or so, or a little more than that, I guess, nobody was willing to accept that they were important pollinators. It was remarkable. I go to meetings and you take pictures like this and show them to people, and the bee people say, well, there's no evidence that they're pollinating. They're just picking up pollen. That's all. And that's really changed in 20 years. There's been a lot of research on it showing that, yes, they are delivering pollen, they are pollinating plants. And in fact, they're probably almost as important as bees in terms of agricultural pollination and probably global pollination in general. So of course, there are lots of fly groups that are important pollinators. Their mussids are really important, tachinids are really important, but syrphids are certainly right up there and pollinate a lot of our food crops and whatnot. So they're great for somebody working for agriculture because they're agriculturally relevant. In addition to this general, almost uniform behavior across adults of being nectar feeders and pollinators, there are some other interesting behaviors that are pretty common in the group. Many are hilltoppers. If you've ever gone to hilltops looking at insects, you'll find certain groups are prevalent there. Parasitoids in particular are overwhelmingly common. So tachinids, pipenculids, things like that are common on hilltops. But syrphids, a lot of syrphids go to hilltops as well. And these are male insects going to hilltops, staking out a little bit of turf there and waiting for a female to come up to mate with them. And it's not an obligate behavior. Typically, what'll happen is it changes depending on populations. So in years when an insect species is rare, probably all the matings are on hilltops because the females can't find the males otherwise. In years when you have a boom of one species, the females will find males off the hilltop. And so it's an evolutionary stable strategy of mate finding. Sometimes it pays to go to a hilltop, sometimes it doesn't. And it's probably species that have mobile hosts and food resources that are hilltoppers the most. So parasitoids are heavily represented on hilltops. So if you work on, I don't know any hymenopterus that do hilltopping, but man, there's a lot of microhymes on hilltops. So parasitoids go to hilltops. And syrphids that are plant feeders tend not to go to hilltops, but syrphids that feed on mobile food resources like aphids and things are often hilltoppers. So it's a big thing. A lot of syrphids are also migratory, and there's been a lot of research on particularly Episurphus balteatus in Europe, because there are billions across the English Channel every year in migration. You can see it on radar. It's really cool. Here in North America, almost nothing known about migration. We've got a little note my son and I are writing on Aristolus tenax, common dronefly, and Halophilus fasciatus, the narrow, what did I call it? Narrow-headed marsh fly. And those two, we found thousands and thousands migrating this fall along the north shore of Lake Erie on a day when there was lots of bird migration also going on along there. And they were migrating dragonflies and other things. And last year I documented a big wasp migration along there. And Sister Circus, a group of wasps, were moving by the tens of thousands. So we still don't know a lot about migration. There's lots to learn there. So that's a fun thing for any of us who are out to look for. So what else about natural history? So let's talk about what they feed on as larvae, because that's what's really, really variable. I can only think of one other group of Diptera that has as Catholic a taste as syrphids when it comes to larvae, and that's Phoridae. Phoridae do almost everything known to insects as larvae, and that's almost as diverse in syrphids as well. So everything from plant feeding to fungus feeding to parasitoids, predators, the whole 9 yards, a lot of saprophagus species, and aquatic, terrestrial, you name it. So let's look at a few of the groups. Microdons, the ant flies. These are ant predators. They live in ant nests, and we don't know a lot about them. Only 8% of the hosts are known worldwide, so very, very poorly understood so far. We're looking at methods to ramp that up by perhaps sequencing the cuticle, trying to get environmental sequences off the cuticle of adult microdons that are in collections, and see if we can match that up to host ants that they've probably come into contact with. So hopefully we can boost that from the 8%, but it's really cool, the work that's been done on them. the larvae have pheromones that mimic the ants. And so the ants just think they're other ants. And they go around feeding on eggs and larvae in the nest. And then when they emerge from their puparium, the adults don't have that pheromone and they get attacked by the ants. So they got to get out of Dodge quickly. So pretty cool. So here's some Microdons. They're a really, really diverse group in terms of morphology, colors, spectacular antennae on some of them. Crazy, but so hard to find because they're ants associates. They don't go to flowers. They're not big hilltoppers because they know where their resource is, right? So they're going straight to the resource, to the ants. So they're often extremely local, right around the ants they're attacking and nowhere else. So lots to learn about Microdons. Menno Reimers, the man that works on those, he's the world expert on that group. The Arist Lines, as I said, is a paraphyletic group. They do lots and lots of different things, but even once we straighten out the monophily, the Aristallinae sensu stricto do all of these things too: Phytophagous, Saprophagus, Predaceous, the whole 9 yards. You can see the other subfamilies we're going to raise in here. Middle row, left and center are Volucella and Copistylum. That's going to become the Volucellinae. These are Volucella live in bee nests as predator, or feeding on a lot of debris mostly. And Copistylum are super abundant in the tropics, often in bromeliads as larvae, filter feeding. The other subfamily down in the bottom row there, Syrphinae, terrible name, awful to pronounce. They're often these wasp mimics, thread-waisted things. And the other group, don't have it shown here, is Humerus and Merodon. These are the plant feeders that are in their own subfamily, Merodontinae. So we're going to straighten that out shortly. In terms of life history, you can pick out certain groups, certain lineages that do certain things, but then there's all these things that are wacky and go off and do their own. And again, this is how come Surfids are so plastic as larvae? It's crazy. So this one, Nepenthes surface, is in pitcher plants. You've probably heard of Wyomia, the mosquito in our area that's in pitcher plants, lives in there and feeds on carrion that falls into the pitchers. Same thing with this Syrphid that lives in, this is in Indonesia and New Guinea. Syrphinae are mostly predators. So this is a huge group with thousands of species, and mostly are predators of soft-bodied things like aphids, but also scales and stuff like that. But then within the group, they walk off and do all kinds of crazy stuff. So here's what they look like as adults. Pretty consistent overall. But here's some of the weird stuff that has been found. They're not all predators in that way. So this one was found. It lives in bromeliads, so it's more like that volucelline that I described, that they're in bromeliads. Very, very, very different than all other surfines. And this is just like cropped out in the middle of the phylogeny. This one, people were seeing them in areas with honeydew, with whiteflies, and thinking that they were probably feeding on honeydew. Nope. They're actually attacking flies that come to the honeydew. They're feeding on sarcophages and tachinids and things that come to the honeydew. So crazy predator. This was a cool one, just was published a couple of years ago. Chimo Manguel was involved with this. And some South American colleagues of his found these larvae in the sundews, just like the pitcher plants, right? This is a pretty hostile environment that's made to digest insects, and Toxomers basalis is living on the sundews and stealing the other insects that get trapped in the sundews and feeding on them. Pretty crazy. And in the same genus, Toxomers politis is feeding on pollen. So like, crazy. And we've even got leaf miners within the Syrphinae, a few leaf miners. So really, really incredible how plastic these things are in their ability. Like I also work on pipe and culids, and they all do one thing, right? They're all except one little lineage, all are parasitoids of leafhoppers, plant hoppers, spittlebugs. So that's normal in Diptera. This is crazy stuff that they're all over the place like this. So Pipizynes, I mentioned earlier, are mostly predators of woolly aphids and root aphids and things like that. So again, a really poorly known group ecologically. We only know life histories for a handful. So that gives you a bit of a background on why I'm passionate about Syrphids, I hope, and why I think they're an interesting group to work on. I'm just going to skim over a bit of the work that I've done with different colleagues in the group. I've worked on many different groups of insects over the years, and I've never met a group of people like the Surfid group. There's about 100 passionate Surfid workers in the world, and we have meetings every couple of years. And we all work with each other. It's extremely collaborative. I've never seen such collaboration as occurs in this group anywhere else. It's amazing. So a lot of these things are done with other people, and different names will pop in and out. But anyways, this is a student of mine, Kevin Morin's been working on Cryorhina and relatives. And this genus Sphecomya, he revised a couple years back with me, and they're really spectacular flies. They're wasp mimics. Almost all of them are perfect mimics of wasps. But as often happens when you do a revision, you find out some cool things. So at the base of the group, sister to the rest of the Sphecomya, is this thing in the right-hand plate, bottom left, that's a bee mimic. That is Sphecomya metallica. And we didn't know that was Sphecomya before Kevin tackled this group. It was placed in another group. It looks like a Cryorhina, being a bumblebee mimic. But in fact, just like the larval natural history seems so plastic, mimicry is extremely plastic. And often you'll find groups where wasp mimicry re-evolves multiple times, or bee mimicry re-evolves multiple times. It must be in the genetic ground plan for these things to be able to pop in and out so much like they do. Within the Petonii group, in the top left picture here, you can see a couple that are not like the others, and those are stuck in the middle of the Petonii group. So you can get these really divergent jumps in terms of morphology. And it's one of the reasons I really like working on phylogenetics of Surfids, to see what's going on with mimicry, what's going on with larval life history, and how are these things popping up over and over again in terms of evolution of the group. This is another group that Kevin and I worked on, another group of Cryorhina, and Romalia surface was only known from a couple of species in the neotropics, and we found 10 species, 7 of them new. They're all pretty much like this painting, very, very spectacular, perfect mimics of bees. Other groups I just thought I'd throw out. I've done tons of revision. I've just thrown in a few examples. This is a fun one because it's a really good honeybee mimic. This is Afrosurface. We thought there was only one species in Africa. We were collecting over there, and we found they weren't hilltopping, but it was the same idea. They were landmarking. There were males all hovering in one spot, sort of under a tree in a sort of semi-opening along a trail. And turned out that it was a second species, and we figured it out and published that. But just a fun group because it's such a good honeybee mimic. This was a group that I worked on with one of my grad students, Andrew Young, who's now a professor at University of Guelph. Andrew worked on Australian salota, and salota in North America and Europe are really boring black flies, and in Australia they look like this. There are some black ones, but most are metallic greens and purples and whatnot. Pretty fantastic. So there were 26 new species out of the 34 that we treated. So I love Australia. It's a candy bag, right, for entomologists. There's just so many new things to find there. And it just seemed like every time we go collecting, we'd find new salota. We don't even know what these things do as larvae yet. We're pretty sure that they live in wet, rotten wood, and they're probably sapropores in there. But and we only know that because there's pictures on iNaturalist of them ovipositing in locations like that. So fantastic flies. So what other revisions am I up to? Those are all published. I also work on pipenculids, and I'm doing lots of revisions of those with Benam motamedinia, the postdoc of mine in Ottawa. And a big paper with him and Mihai Folvari we're working on right now is Thomas Variella of Australia. So the bottom right picture there's Thomas Variella. And to put it in perspective, there's about 70 described species of pipenculids in Australia. We've got 104 species in this genus, 100 of them new. 100 out of 104 are new. So pretty exciting. I'm not going to be doing a field guide to them. The only way to tell them apart is genitalia and some very, very minuscule leg characters, basically. But it's like opening up Christmas presents, taking the genitalia out of these things. Like, oh, another one, another one. So lots of those. Another one I'm working on right now is triglyphus in Australia. I'm doing a few Australian revisions. That's the bottom left, kind of dumpy for Syrphids, not super colorful, but there was one species thought to be widespread in Australia. It turns out it's 15. So classic Australian story. Other revisions we're doing with Kevin, Cryorhina, Humerus, a whole bunch of us working on some Humerus revisions worldwide. Again, it's extremely collaborative, this group of Syrphid workers. Temnostoma with some European colleagues and various others. So that's the revisions. I thought I'd talk a little bit about phylogenetics too, because that's the other bread and butter of my research. Of course, you know, we're using different methods and always changing and growing with what we know and are learning and trying. We've done multi-gene phylogenies for years of different groups, from micropesids to pipenculids and Syrphids and you name it. Then we tried playing with anchored enrichment, and I'll talk about some of our publications and things we've done. That's what the lemons pioneered. And then more recently, we're working on hybrid enrichment. I'll tell you a bit about that too. And I'll profile mostly Syrphid work just because that's what we're mostly doing. So this was a paper Kevin Morin led. We spent years collecting this multi-gene data set in my lab, and Kevin took it and ran with a data set. And it turned out that I think we're using 5 genes, and those 5 genes told a story that's remarkably similar to a hybrid enrichment analysis using 1,300 genes. So you know, pretty good. I mean, some of the backbone supports weren't as good as they are in the enrichments phylogenies, but still, he was really interested in the clade at the very bottom here, which is the Cryorhina group, and that's really what we were after. And we got fantastic supports all through that group. He got 100% supports through that, so that was his goal. And then the rest of the phylogeny was almost like all outgroups, right? And it showed us that we have this paraphyletic Arist Line group and whatnot. So and we see that crop up over and over. So Andrew Young did a paper using the lemon data that we generated, this was a very, very small data set, but told us pretty much the same thing as we saw in Kevin's more recent multi-gene paper, and the same thing as we're finding with our hybrid enrichment paper. So if you go to ESA and you have a chance, look up Kevin Morin while you're there. He's doing a talk, and he'll bend your ear and tell you all about this stuff, but it's pretty exciting. Chimo Manguel's lab in Bonn, Germany, developed a new set of probes specifically for Syrphids. And we use those probes. We've now got sequence data for almost every genus of Syrphidae and many, many exemplars of each genus. Pretty much 100% support across the board. We're really, really, really getting a great idea now of the phylogenetics of the family as a result. But what's cool is we sequenced some other stuff. I had some project money at work, and they wanted us to sequence everything from the collection. It was genomic characterization of the collection was the project. It was a boondoggle, typical government. Anyways, so we decided, well, what the hell, we'll try it with this and see if we get any results. And those probes worked really, really well across all Diptera. And so Kevin's been working on a Diptera phylogeny paper using these probes. And we've got other colleagues working on other families using them. And Kevin's developed this amazing pipeline. He's taken this pipeline and it's developed for this data set that we're working on, but he's reanalyzed other data sets that have been published in Diptera, and he's getting 10-fold or more increases in data from what's been published just last year, from what was published a year ago. So this is an awesome new pipeline he'll be talking about that he's developed. It's pretty cool. So we've done the same thing with pipenculids. This is a multi-gene phylogeny I published with Benam, Motamedini, and a couple other pipenculid colleagues. And we've now got a target enrichment phylogeny that's looking very, very similar coming through here. And the big problem with pipenculids is in the genus Eudoralus, which was just a hodgepodge. And so we're trying to, you know, one of the big goals with this for me as a taxonomist is try to break down the classification, name new genera, and make manageable bits for revision, because right now Eudoralus is like 1 out of every 3 species of Thomas pipe and culid in the world is a Eudoralus. And if we can break it down and define small monophyletic groups that are more manageable, I think we'll get more done taxonomically in the group. Other groups I published with Sean Winterton on his great paper on Neuroptera phylogenetics. I worked with the Odinate team, Bybee et al., and did a paper on phylogenetics of Odinates. This was a really fun paper, brought in, of course, timing and a lot of fossil stuff. It was cool to work on. So anyways, I really enjoy playing around with phylogenetics. So that's been a lot of what I do. But I wanted to finish the talk about really talking about something that is more novel that I led. And probably everybody in the room is also a naturalist, or at least interested to some degree in natural history of other stuff. And you've probably used books like these over your careers: bird books, wildflower books, mammal books, whatever, field guides, right? And there are guides to insects, but within Diptera, there was nothing comprehensive for any group of Diptera as a field guide. There was this book, Britain's Hoverflies, which is fantastic, but it's not comprehensive. So it gives you a picture of what's there, but you don't know for sure that you have species X, because maybe only half of the species are shown for each genus, right? So it's not comprehensive. And I wanted to do something that was comprehensive. So in 2019, we published this in Princeton University Press with them. And it's a field guide to the Syrphidae of northeastern North America. So if you haven't seen it, I hope you check it out. It covers this part of the world, so your area is covered. Why this area? For example, why isn't Manitoba included? Because it should be by the looks of this map. It's because it has to be a damn field guide. And if I put in Manitoba, it adds 70 species, because a lot of Western stuff occurs in Churchill. It's crazy what's in Churchill, which is up on Hudson Bay. So there's a whole bunch of Western stuff gets in that makes it difficult. And obviously, if we go down into Tennessee to Florida and whatnot, it goes crazy. So we'll do another field guide maybe for the Southeast at some point, because it's actually pretty straightforward. There's a lot fewer species there. So that's the coverage area. This is what it looks like. It looks like any field guide to birds that you've ever picked up. Well, I would argue it looks like the best field guides to birds, because we've tried to incorporate all of the really good features of field guides I've used over the years. So you've got the Peterson sort of arrows pointing to your field marks. We've got photos of, dorsal, lateral, and whatever else is necessary to show you the characters. We have these maps that show dots where there are locations, but also predictive colors as to where we think the species occurs. The silhouette is actually life size, and the eyeball, we have an eyeball, we have a hand lens, and we have a microscope. And that tells you, eyeball means you can field ID it. Hand lens means you're probably going to have to catch it and look at some minute characters, but probably can release it when you're done. People like to do that, right? Naturalists. And then microscope means you're going to have to kill it and take it home. And only about, oh my goodness, it's not many, like 20 or 25 species require a microscope. Most of it you can do with a net and catch and release or whatever. So another thing we have is we show a hilltopping symbol on species that are known to hilltop. So that's what it looks like. How did we do it? It's not simple like I don't want to denigrate people working on bird field guides and things, but I could write a bird field guide by going and doing all of my research on the internet, right? And we had to do a lot of novel research for this, and I'm hoping others in this room listening to this talk will do some field guides. I don't want to take away from how hard it is, though, because it's going to take some time. So the first thing is mobilizing data. So I had teams of summer students for many years would follow me, and I'd identify Syrphids in collections, and they database them. And this is a picture in the bottom right at Chris and Betty's home in Florida with a team of summer students I took with me. And we worked to get the data into databases. And now, there's been big changes since 2019 when I published this. Tons and tons and tons of data are now on iNaturalist. And so you can harvest a lot of that data, check the photos, see if they're correct, and include those data as well we did include iNaturalist and Bug Guide data in our guide, but it was pretty minuscule compared to what you could do now. So Victoria Knoll was one of my summer students, and she worked really hard to model all that data using Maxent to create these predictive maps. That was a huge job and took 3 iterations before we had it right. And of course, I had grad students working on revisions because there were whole groups that were untenable, unworkable in a field guide until they were revised. So Michelle led the DASI surface revision as part of her master's. Andrew revised Platychirus as part of his master's before he went on and did the Australian stuff and phylogenetics for his PhD. And we did others. I worked with Chris Thompson on Sarcomaya and a few other revisions. So all this had to be done to set the stage before we could even do a field guide. We couldn't have done it without Chris's Conspectus. So anyone who knows Chris Thompson knows he loved his Conspectuses, Conspecti. And so he would do these, a summary of everything known as a Conspectus. What do we know about group X, and throw it together into a Conspectus. And Vacheroth provided what we knew, even though a lot of it was unpublished. And so the two of them worked well together. They both shared information. Dick shared all his keys and things with Chris, and then Chris shared it with the world in this unpublished Conspectus. And that really, really helped us get a lot of the work done for the field guide. Now, unlike most bird guides, which just present what we know at the time about birds or, you know, whatever the field guide is focused on, this had a lot of novel research in it. So Michelle and Kevin and I looked at DNA barcodes for almost all the species in the field guide, and we revised species concepts. We changed our generic concepts. We had unpublished phylogenies, so we're able to change some of the generic concepts and whatnot as well. So in fact, we elevated 9 taxa to generic status. We had 1 new generic synonym, 16 species resurrected from synonymy, 20 new synonyms, and 18 undescribed species were treated. So it's like a huge research paper as well as a field guide. Most of the field photos were taken by Steve Marshall, but I managed to get 65 other photos that we really needed from Bug Guide and iNaturalist and places like that. And all those folks contributed them for free. They just got a free copy of the guide for their efforts. And that was great. And then how do you test a guide like this? So Chris and I ran 2 courses on Surfids in Ottawa, and we had tons of people. We had a big grant studying pollinators at the time, and they all came to study Surfids with us for a week. And they were our guinea pigs, and they tested the guide, and they also tested an online key we put out. So one of the things this field guide does not have is it does not have a key. And a lot of people find that uncomfortable when they're dealing with insects, they want a key. But I tell you, I learn birds by flipping through the guide. And that's the best way to learn as a naturalist, I think, is sit every night, flip, 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 flip. And doing that with the Surfid guide, you're going to learn them faster than farting around with a key. But we put a key online and published it just so people could use that as well. And we linked to that in the field guide. So, and that's typical, right? It's dichotomous and takes you through to the different genera. So anyways, to finish up the story, you know, I'm a government worker, and that creates all kinds of interesting problems when you write a book. Princeton University Press could not get their head around it that I couldn't receive any money from said book. And so that was confusing for them. And of course, they had a legal team, and Ag Canada had a legal team, and they didn't talk together very well. And so there's a lot of confusion and whatnot. And there's a woman, Stephanie Warner, that worked in my building that really sorted out a lot of this. But I'll tell you, when you work for the government, you need every level of authorization, right? It goes way up the food chain, and yeah, even the Queen has to have a say, right? So not really. Anyways, and then once it's all done, we had a couple, anonymous reviewers, typical of entomology. Thanks, John Klimko and Martin Hauser. Not very anonymous, right? You always know who's reviewing your papers, right? It's just, anyways, they did it's a huge job to vet a guide like this, and they did a fantastic job. And then we had a bunch of other people reviewed as well for us. So we had to incorporate all that. And then I'd done everything in InDesign, prepared all the layout before I ever went to a publisher. And because I didn't want the publisher changing anything. And so Princeton was fantastic. Rob still took my guide and didn't change anything about the layout, but made it pretty. And he really made it beautiful. And we had an amazing copy editor. I still haven't found hardly any mistakes in it. She did an amazing job. So anyways, the whole process was a lot of fun. I highly encourage anyone. Robber flies is way overdue for a guide like this. Bee flies, lots of beetle groups. You know, if you're interested, talk to me about it, and I can walk you through some of the process. And it's not nearly as onerous as it was a few years ago, because so much of these data are mobilized online now compared to when I put this together. But anyways, lots of things to do, lots of plans, and I'm going to leave it there and take any questions now. Thanks very much.
Hey Jeff, this is Mark Metz.
Hey Mark, how are you?
Good, thanks. Good to see you. Good to hear from you. Great talk. Appreciate it. So on your tree, your text was pretty small, but I think I still saw Chrysotoxum and Doros still in Surfine.
Yep, you bet.
So has anybody done any work to try to explain why so many of the high fidelity mimics are in Arist Lines, while Chrysotoxum and Doros may be the only high fidelity mimics in Surfines?
No, and I think that's coming once we publish we've got 2 more big phylogenetic papers to come out. And once they're out, I think some of my colleagues that work on mimicry, like Tom Sherratt, I think they'll take a run at some of those big questions, because I think then we'll have all the fodder we need for them to really dig into it. But it is fascinating, isn't it? That one subfamily really, it's all going on there. We did a fun little paper. It's actually my only Nature publication, with a student at Carleton University, and she was trying to figure out why imperfect mimicry arose, because it doesn't make a lot of sense when you look at it. And she found that mimicry was driven by birds, by vertebrate predators, not insect predators. And the bigger the insect, the better mimic they had to be, because birds will take a second and third look if it's a high value meal. And so the imperfect mimics are mostly small. So it may be that simple, that the Syrphidae tend to be smaller, and that's why they're mostly imperfect mimics, right? Chrysotoxum's a big beefy Syrphidae, and so they've evolved perfect mimicry. So that would fit what Heather discovered anyways.
Jeff, I have a mimicry question for Sound. I was recently involved in looking at some Anastrapha. There were species described that look fairly similar, but they're sort of different colors. Like one was basically brown, another was more was yellow with brown marks. And somebody reared them, and they both came out, and CO1 looks all the same. So it looks like there's different morphs. Do you see that with Surfids, or do they just tend to mimic, each species mimics one model?
Yes, it's a great question. So yeah, we do see that. Kevin's that was part of his thesis, looking at cryorhines, and it's kind of cool there. So I'm going to start there and I'll work up to your question. So he found there was one lineage of cryorhines currently under the genus Cryorhina. He's going to name a new genus for it because it's a monophyletic group, and they're only queen bumblebee mimics. And the other group, they're worker mimics. And there are some species that have 2 color morphs, and they might be a worker mimic or a queen mimic, or their bees have 2 morphs and they're mimicking both morphs. So within Cryorhina, you get a few of this sort of double morphs, and we checked it with DNA barcodes, and we've got some rearing data as well, and it seems legit. Within the genus Volucella, which are perfect mimics that are in bee nests as larvae. It's really complicated, the taxonomy. They're really a bugger to work out. And I've not yet published a revision that I've had sitting around for way too long, because there are multiple morphs in one species, for sure. And I think I've got enough data now that I've finally worked it out. But for example, if you look at Volucella bombolans, bombolans has 39 synonyms. And the type, if you look at the type photograph, it's online, it's a black fly with an orange tip to the abdomen, like some of the bumblebees in Europe. And there are morphs that are yellow and black, and there are morphs that are all black. There's about 4 or 5 different color morphs of that species, and that's why there are so many synonyms. But it turns out that I now have found that in North America, there's 3 species that were synonymized that aren't synonyms. So it's difficult to tease it apart.
Is that something you can only solve by having some molecular data?
The molecular data was crucial. We couldn't have solved it. I mean, they partly solved it using rearing, in Europe, but really getting to the bottom of the molecular data was critical. But the barcode, the branch lengths are pretty short, which, it would really probably should use multiple genes to sort it out. But the barcodes were enough. It's just, not perfect.
Hi, Ed Barrows here. Am I muted?
Nope, we're here.
Oh,
Yeah.
okay. I really like your book. I'm holding it up. And I'm wondering if anybody is working with actual birds and flowerfly mimics nowadays to see what the behavioral responses are. I know Lincoln Brower worked with butterflies, but I don't know that there's been many papers with syrphids and living predators. Is that true still?
Living predators, probably you're correct. Heather's paper, she used a stuffed blue jay and used that to elicit responses and whatnot. So yeah, using living birds, it would be a challenge, but I think is open and would be really, really interesting for research.
Yeah, because I once tried that with tortoise beetles, and the birds could tell them apart. Naive birds could tell the tortoise beetles and the ladybird beetles apart really easily. There wasn't any problem for them.
Cool. That's neat.
There's a couple of comments and questions in the chat. See, George Foster was complimenting your book. He says, "This is a fantastic book. I use it all the time for syrphids I collect all the Coropids."
Cool.
So,
Thanks.
Latana says, "Great talk, Jeff. Just wondering, what is the diversity and phylogenetic status of Graptomyza?"
Okay, good question. So Graptomyza is in that Volucellinae new subfamily we're going to propose. So they're related to these bee nesting Volucella. Graptomyza is an old world group, and I would say it's one of the most poorly known genera in terms of taxonomy. So there's lots and lots of undescribed species in the group. They're probably like their New World counterpart, they're probably saprivores, and they're probably in bromeliads and things like that. To give you an example of Copistylum, which is the New World equivalent of Graptomyza, there was some work done in Costa Rica, and I don't know, there's like 50 or 60 species of Copistylum were known from Costa Rica, and they reared 200 species from bromeliads, just like in a fairly small project. So like the diversity is almost unbounded in this group, and it's all undescribed. And Graptomyza is probably not quite that crazy, but it's probably close.
Any questions?
Jeff, this is Bill Murphy. I'm going to try to share the screen here. Oh, it's disabled by the host.
Well, let me see here. I think maybe Jeff has to stop sharing his.
Here, let me stop my share.
Do multiple.
Okay.
Try it now.
You have to give them permission.
Still disabled.
Where do we do?
I think you do. I'm sure. Just do.
Share screen share. That's true.
After all this, it's a human I'm going to show you a picture of. Anyway, I guess it's not going to work. It keeps saying host disabled participant screen. You mentioned a Victoria who's working with you. There was a very young, very fast labeling person at the CNC in 2012. Is this the same Victoria?
This is the same Victoria. Yeah, she worked with me for I don't know, 4 or 5 summers, and I've never had a summer student as efficient and accurate as Victoria's. She's gone on, she's doing some policy stuff with that's her, yeah. She's doing some policy stuff with the government now. She working on Siomyzids there, Bill?
She was kind of assigned to me. I was identifying your unidentified Siomyzids, and she was, and probably still is, the fastest at working with labels, putting on barcodes. I'd never seen anybody who kept turning to me and saying, do you have any more? It was just, it's kind of person where you think, if I could hire you away from CNC, I would do it. Yeah. So I'm glad to hear that she's still with it. Lucky guys up there.
Yeah, well, she's working on policy now. She doesn't work with us anymore, but they're lucky. She was an amazing summer student, and that's why I could afford to give her to you to work with you for a couple of weeks, because she got so much done with me the rest of the time. It was like just hard to keep up with her.
Yeah. Well, you can go and tell her she's internationally known now.
Okay, I will.
Okay, let's see, any more, this, or great talk, thank you, etc.
Yes, I have a question. I'm Juan Perilla, and I saw that you showed the Microdon tini. You show a picture of one of those that had the antenna branched, and I was curious if that's a character that has shown up several times independently in the family. If there are more Syrphids that have this trait, and also if you know anything about what could be like the driving forces for that kind of antenna. Because that's something that has appeared multiple times in the clinics, but we know nothing about that.
Yeah, it's a great question. So I would say it probably hasn't occurred to my memory. I can't think of it right now outside of Microdontinate. So only the ant flies have that, but it's occurred multiple times within that lineage. So again, some kind of underlying genetic ground plan perhaps, but anyways, it's popped up over and over. And then there are lineages where there are several species have it. Like within the tachinids, we're not sure about what it's for. One would speculate that it's something to do with mate finding, but I think it makes more sense. It's something to do with ant pheromone detection, but who knows? There's so many questions. You know, it's like any group of Diptera, right? Or any group of insects. There's just so many questions that could be asked. It's endless. And we're hoping the phylogenetic framework really helps people dig into some of those questions. And having this new pipeline that Kevin's developed, we're hoping that other labs can very, very quickly and easily ramp up an additional node of the phylogeny. So if they're interested in ant flies and they want to look at one group, then they can just, quickly work up a couple of kits and get the data and incorporate it into the tree. So lots to do still. Any other questions?
I think that's it, yeah.
Okay. Well, I just to finish up before I go, I can't say enough about my colleagues that I work with. And if you ever get a chance to go to one of the Surfid meetings, Rob Ruffley was a coleopterist, and he always came to the Diptera meetings, and there was nothing wrong with that, right? It was good fun. And you don't have to be a Syrphidologist to come. It's a really nice atmosphere, and they're usually in interesting places. This year's was in France. Anyways, please come. If you see my students at meetings, Kevin at ESA, please engage with him and talk to him. He'll tell you all about this. He knows a lot more than I do about the whole bioinformatics side of things. So anyways, thanks for having me tonight. It was a lot of fun talking to everyone. I just wish I was there in person.
Thank you, thank you.
Almost a perfect.
Yeah, absolutely. I was actually going to say that it was great seeing Chris Thompson again. And he contributed a lot to our society. Of course, he was with Systematic Entomology Laboratory. And one of the traditions of our society in our journal is to publish a little obituary. So perhaps you and I've been trying to get, well, maybe, but not trying, but I know there's one coming out, but it would be great to have a little something about Chris in one of the proceedings.
Well,
Yeah, that would be nice to do. We have a whole issue of Studia Dipterologica that's devoted to Chris that we're putting the final touches on this week. So that will be out this year. And it's got, I don't know, 15 to 20 papers in it, all of them with a Chris angle and some with some Chris stories. And so it should be really nice. So watch for that if you knew Chris Thompson. Don't worry about the fly stuff. Just read through and look for the juicy bits about Chris and, you know, et cetera and so forth.
yeah.
Et cetera and so forth. Is that how you named it?
We should have. I mean, that was his catchphrase, right?
Uh-huh.
Yeah, and I think you guys have a lot of kind of litwax images are going to be on the cover.
Yeah, that's right.
And you know, you were mentioning about your community, but I don't know if everybody knows, like Jeff came down for 2 weeks this summer, just went through, it was almost a whole row of cabinets of what Chris called his research material. So it was a mix of loans and stuff that you know here. Jeff went through a big part of it and sorted it all out. It's very generous with his time. And he took a whole van full of Chris's manuscripts and notes back, and imagine you'll be looking at that for a long time.
Yeah.
Anyway,
Hey, Jeff,
Hey, Mark.
are Gilbert and Rotheray still active?
They are, yeah. Rothray's retired and pretty low key now, but Francis is still going strong.
Great, so we've reached the end of our meeting. We don't have to go, but I'll go ahead and adjourn. If there are any final announcements, I saw that there was some links. Thank you, Mark, for posting those for the ECN and the ESA. Definitely make sure to check those out. Okay, so I'd like to ask for a motion that we adjourn the meeting. Oh, and then the next meeting is December. Yes, and again, just yes, for sure. So now you can hear us. So here we go.
Over 50 people combined.
Oh, wonderful. So yeah, so about 50 people combined. And,
Almost 60.
that's great. So, okay, well, thank you very much for the talk and for attending. And we'll see you next month. And there'll be elections next month. So, okay, so I'd like to ask for a motion that we adjourn the meeting. Okay, second. Okay, so all those in favor?
Hi.
Okay, the motion passes, meeting adjourned.
Hey, thank you. Good night.